Micro Nano Biofluidics group at IIT Madras. Welcome to our monthly talk series. Just a quick note before we introduce our speaker and proceed to the talk. You can ask your question by typing in the Zoom chat box, and we'll moderate the question. The speaker will respond to it at the end of the presentation. We are extremely delighted and honored to have Professor David Weitz from Harvard as a speaker for the opening talk. Thank you, Professor Weitz, for accepting our invitation despite your busy schedule. As such, Professor Weitz doesn't need any introduction. He is a very well-known and highly accomplished researcher in the field of physics and science. He is a pioneer in the field of colloid physics, biophysics, and microfluidics. And his work covers fundamental science to direct applications in industry. Interestingly, he is also a part of the popular lecture series and a book on science and cooking that nicely explains and solves culinary problems through science. His today's talk is on the topic dripping, jetting, drops, and weighting, the magic of microfluidics. With that, we welcome you, Professor Weitz, to deliver the talk.
Okay, well, thank you very much. It's a pleasure and an honor to be here. And it's always nice to hear of a new group that's getting started in exciting fields. Of course, I'm biased. I think this is an exciting field. And I want to give you a bit of an introduction to work that we've been doing over the last many years, I guess. Describing some of our efforts to learn about microfluidics and to make it useful, to understand the physics and to do something with it. So the work I'm going to tell you about is really the work of many, many people. I've listed as many as I've had room for here. They're all former postdocs, graduate students, and visitors to the group over the years. Many of them continue to work in some way, shape, or form on some of the microfluidic concepts, all of them doing very interesting work wherever they are. And I'm going to tell you about work that we did together in the group, trying to learn something about microfluidics. And really there are two themes. Most of my talk will be talking about a material science theme. It's making materials with microfluidics. But that's something that we like to do. We're very interested in. I'd like to try and convince you that it's a worthwhile thing to do, and it's something that you can do. You can actually do in surprising ways that lead to interesting applications, utility. But really, I think the more common and probably the more important long-term use of microfluidics of the sort that I'm going to describe is in what I would call sort of lab-on-a-chip applications. And I do want to describe some things with that as well. So this is very much of an overview and an introduction to microfluidics as we practice them. I think before I even start, what I'll tell you is that our theme is really to use multi-phase flow, to use drops rather than continuous fluids. And I like to try and convince you that that's really a very powerful sub-corner of the field of microfluidics that has many important applications and a lot of really interesting physics. So, on the materials science front, I'm going to describe the synthesis of new kinds of particles. I call them colloidal scale, but typically they're somewhat larger than colloidal scale. They're in a scale of microns, maybe submicrons, microns to hundreds of microns, but they're in a scale that has overall a very important applications and a lot of really interesting physics. So typically, to make these kinds of particles, it's done as a batch process. That is, you take a bunch of things, you mix them together, and you stir them, or you do whatever other kind of processing you would like to create many large quantities of small particles. Now, to do this, because you're just mixing things together, the way you make them, what you get, how you do it all is very constrained by just the way the chemistry works, by the bulk chemical preparation. In particular, if you want to make absolutely uniform sized particles, very monodispersed particles, you have to do that by controlling the way the chemical reactions work, the way you mix things. Everything is done on a bulk scale, and it's actually very difficult to make things extremely uniform in this fashion. You can do it with a lot of very tight control over a lot of different parameters, not all of which you really understand, but it makes things very difficult. And so I want to describe, by using microfluidic techniques, a new route to particle synthesis. It's a continuous process. That is, you don't make it batch by batch, but you make things all in a continuous fashion. The difference is, in this case, because you're using microfluidics, you make each particle one by one by one by one. Everything is made one at a time. And that gives you really, truly exquisite control over each particle. You can adjust things so everything to each particle is done exactly the same way. It becomes very simple then to make things identical, to make things very, very monodispersed. It's a very simple thing. It's very easily achieved. And the way we do this is by making drops of one fluid in another, or by making emulsions. So an emulsion is nothing more than drops of one fluid in a second fluid. Typically, the two fluids are immiscible. They don't always have to be, but for most of the things that I'll describe today, you can imagine two immiscible fluids. And I'm going to show you that with the microfluidic technology that we'll introduce, you can make very, very monodispersed, very, very uniform drops. Again, you're making the drops one at a time, so each one can be exactly uniform. But now, to add some sophistication to make really interesting new particles, each drop will be used as a template. That is, you'll make something and you won't just use the single drop itself, but you'll build things around it. You'll take advantage of the way you can control the fluids to use the drop as a template to make a more sophisticated kind of material. And I'm going to show you how we can make really new and functional materials using the drops as templates. And to do this, we're going to use microfluidics. We're going to use the really exquisite control that you have by controlling with microfluidic technology, controlling the way fluids mix, control how much you mix them, control how you structure them. Everything will be under really precise and very tight control using microfluidics. So microfluidics really, to me, there's nothing fancy about microfluidics. The goal of microfluidics is just to use micron scale or sub-millimeter. It doesn't really matter. I don't want to overburden the terminology. We call it micron scale, but we're using channels that have exactly the same dimensions, or approximately the same dimensions, as the fluid structures that we want to make. So we're mixing things by controlling the way you mix them, by controlling the channels in which the fluids flow. Now, one thing you should ask right at the start, and you should be concerned about, and this is, in fact, if you think about materials production, this is probably what has always limited the applications of materials production with microfluidics, and probably still does. And the issue is, can you make enough material that it's actually useful? That is, if I'm making things one at a time, one drop at a time, but I really need billions to trillions of drops, can I possibly make enough material that it's actually useful? And I'd like to try to show you that, in fact, I think you can, and that you can really adjust things, control things in a way, if you're creative, if you think through how you need to do it, you can make things, you can make enough material that it's actually functional, that it's actually useful. So how do you make droplets? How do you make an emulsion? Well, Anderson Shum, one of the early students who worked on this, he's now a professor in Hong Kong, he drew a little schematic of how you normally think about making drops. So imagine you have two fluids that don't mix. That's the way you make an emulsion typically. So imagine you have oil on the top here and water. Oil and water don't mix. What do you do to mix them? Well, Anderson says, you shake them. There you go, you shake them. And if you do that, you end up with something that looks like this. These are drops, for example, drops of oil in water, if you're trying to make a vinaigrette or something like that. And you can see they're all different sizes of drops. And the reason, of course, for having so many different sizes of drops is that you're just very crudely mixing it. This is this bulk processing that I told you about in the beginning. What I want to do is I want to show you how you can make them absolutely perfectly uniform, absolutely all the same, make them perfectly monodispersed using microfluidic technology. So for doing this, we wanted to use a technology that would be very practical, very simple, that would be useful for making materials. So we want to use a lot of different materials, a lot of different solvents, a lot of different reagents, a lot of different chemistries. And the standard kind of technology for making microfluidic devices is extremely useful, but is very cumbersome to develop for this kind of application. So we developed our own different type of microfluidic device, which we call capillary microfluidics. These have the advantage that they're very easy to make. They're three-dimensional, so they're easy to control. You can control the wettability, control the way things that are wet, and most importantly, they're very simple to construct. So what do? We take capillary tubes. This is just a vial of capillary tubes. We just go to our friends in biology, our friends in chemistry. They have capillary tubes, lots of capillary tubes. And what you do, they're glass. They're very easy to manipulate. So they're glass in the middle. All you have to do is heat the middle of the tube up and pull it apart. If you heat it to close to its melting point and you pull it apart, you get something that looks like this. You get a very, very sharp tip at the end. And if you control it precisely right, you get a very small orifice here, a very, very small orifice. You start with an orifice that's about 1 millimeter in outer diameter, maybe 0.5 millimeter. So let me write that, 0.5 millimeter in inner diameter. That's shown here. It's about 0.5 millimeter diameter. And you can easily pull it so that you have an orifice of order a micron, down by close to a factor of 1,000. And this is a very reproducible. You have machines that precisely heat and precisely pull the capillaries apart. So if you do it twice, you get exactly the same shape. And so how do you make a drop from this? We do this in a very simple fashion. First of all, we want to be able to align this capillary, this highly controlled, this very sharp tip. We want to be able to align it macroscopically in some simple way. And in order to do that, we take advantage of a very simple idea. That is, we try and put a round peg into a square hole. So here's a square hole, and I align in this around capillary. So this is my capillary here. It's cylindrical in shape. So it's spherical if I look at the cross section. And we take a square capillary that has exactly the same inner diameter or inner dimension as the outer dimension of the capillary. The outer dimension of the capillary is very highly controlled. It's very precise. And so if I have something that's exactly like this, I can align the capillary, the cylindrical capillary, very precisely in this square capillary just by butting it up against two edges. And because it's so uniform, it's completely aligned in that square capillary. Moreover, since here it's a very narrow orifice, I have 4 regions here through which I can also inject a fluid. And so that's exactly what I do, is I flow one fluid through the middle and another fluid through the 4 interstices around the edge, and they come together where they come to a point, and I'm infusing one fluid into a second. And you can see that they're both flowing, both of the fluids are flowing in the same direction. So we call this a co-flow geometry. And this is a perfect way to make drops. Let me show you how we do this then. This is a camera image of the square capillary. This is my cylindrical capillary. I thread the cylinder into the square and I put it all together. And this is what a device looks like. It's held together. As I say, we're very simple. We want to make very simple, easy to assemble devices. This is all held together with our favorite material for holding things together, just some 5-minute epoxy. So here's the square capillary here. The cylindrical capillary is designed here. And these are orifices, or these are tubes through which we can either insert or collect fluids. This is a slightly more complex device, but I'll show you we can make these devices very easily. Anderson, one of my great students, he could make a device much more sophisticated like this. He could make a device like this in about a half an hour. And we're very happy to show anybody who wants to know how to do this. We're happy to share how we make this. It's all very simple. This is what it looks like in the lab. We have to connect the microfluidic device, which is here under the microscope, so we can look at it. We have to connect it to the real world. So we use syringe pumps to inject the fluid through these tubes. We look at everything in the microscope. And this is how it looks like when we do it. I'm going to describe how we make the drops, and I think maybe I'll spend a few minutes and just describe a little bit of the physics of the drop making. And the drop making, the beauty of this drop making is it's done without any huge shear forces. We're going to take advantage of just hydrodynamic instabilities. And for that, I'm going to just digress slightly, take advantage of the ability of my chance to describe a little physics, to describe a little bit of these hydrodynamic instabilities. So this is what it looks like when we're making drops. Here is the orifice. Now you can see it through a microscope. You can see the diameter of the orifice. The inner diameter is about 10 or 20 microns. And these drops are about 30 microns in diameter. I'm looking at them in the microscope. And you can see they're extremely uniform. You can't tell the difference in size from one to the other. If you take the images and try and overlay them, they'd overlay just perfectly. And you can see they're being made sort of, if this movie is playing properly, they're being made about 1 drop per second. Actually, I'm going to show you many movies like this, and they're all taken with a high-speed camera and slowed down. These drops are being made more about 1,000 drops per second. And that's a typical rate, about 1 kilohertz of drops, about 1,000 per second. And you can see they're perfectly uniform. And this should remind you of something. This should remind you of something like a faucet when it's going drip, 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 drip. And we indeed call this the dripping regime of drop formation. Now, if you speed up the flow, this is the inner fluid. If you speed up the flow, actually, let me just reiterate that the inner fluid is flowing here. The outer fluid is flowing in all around it from the outside. So this is the inner fluid, and this is the outer fluid. They're 2 different fluids. In this case, we're making water drops in oil, but you can do it the other way around. You can do whichever way whatever you choose. So if I just increase the flow rate of the inner fluid, then I get what I call the jetting phenomenon. Basically, what happens is the flow rate increases and you get a jet of fluid. It's shown here, but ultimately it also breaks up into drops. You can see, though, now I'm going to try and draw with my pen exactly where the drops are formed. So sometimes they're formed here. See, now they're formed there, but then they're formed at various places. So they're not formed exactly in the same place. They do form drops, but they vary. The location varies from over time. And so the drops themselves, when they're formed, are not quite as uniform as the drops in the dripping regime. So you can understand this. You can understand the difference between them very easily. You can go home, in fact, and do an experiment yourself. Just go to a faucet, kitchen faucet, bathroom faucet, whatever faucet you can find in the lab, turn on the fluid, turn on the flow of the fluid, but turn it very, very slowly, and then you'll get a drip, 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 as shown in this image. If, however, you increase the flow of the fluid, you will see, well, it's just a stream of fluid, but if you adjust it exactly right, you can get it so that there's a stream of fluids, but just before it hits the bottom of the sink, it breaks up into drops, and it does it just before it hits the bottom of the sink. Now, if your control is not precise enough, if it's difficult to adjust, don't do that, but go and take a garden hose, something that sprays water very, very strongly, and let it spray as strong as you want. Let it shoot a stream of fluid out as large as you want, but let the fluid go really as far as it can go. And you will invariably see that before it hits the ground, it will break up into drops. And this is called the Rayleigh plateau instability. And that we can understand the origin of this in a very simple way. So let's go back and look and understand this here very simply. We'll do this simple experiment of using drops in the bathroom, in the kitchen. Here's your faucet, and it flows so slowly that when the drop is first formed, it's still held in place on the end of the faucet by the forces of surface tension. And as it flows, it gets larger and larger. The weight of this drop, the force due to gravity is increasing. That force is just proportional to the volume of the drop times its density times the gravitational force. Gravitational acceleration, it's the mass times the acceleration. That's the force on this drop. It's getting larger and larger, but it's held in place by the surface tension of this fluid against the faucet. Eventually, however, you get to the exact balance where the gravitational force is exactly equivalent to the surface tension force, and then if you increase it just marginally, just slightly more, it falls off, it breaks off. And since it's just a balance of forces, the gravitational force and the surface tension forces, you balance these forces, you get a precise time when the drop breaks. Now, if you have this stream of fluid and you say, what happens far away? Well, you always see it breaks into drops. And the reason for that is, if I just go back here, if I have a cylinder of fluids, it's always susceptible to breaking up into drops. And that's nothing more than saying that the surface tension, which you want to minimize, as the surface tension is the equivalent of having energy due to the presence of an interface. The surface tension is always minimized for any shape if the shape is a sphere. The ratio of surface area to volume is a minimum in a spherical shape. So this shape, a cylindrical shape, is unstable just due to energy considerations to the formation of two drops. And that is the Rayleigh plateau instability. It's saying nothing more that it wants to break up. How can we understand this? What causes the Rayleigh plateau instability? Well, imagine I have a cylinder of fluid and I have a slight fluctuation. So right here, it's slight fluctuation, any thermal fluctuation, any kind of fluctuation that leads to a slight decrease, a slight indentation of the fluid. Then the cross-sectional radius here is smaller than the cross-sectional radius here. That means that the Laplace pressure here is larger. So there's a large pressure here that's driving the two fluids out onto either end. And that causes this thing to grow. This indentation gets larger because you have a larger pressure here that's driving the two fluids out. What's resisting that is the fact that you're flowing with viscosity. So the velocity for the fluid to move in this direction is just given by the surface tension divided by the viscosity. This is the driving force, this is the damping force. And how long it takes to snap off is just this radius divided by that velocity, or the radius divided by the ratio, or times the ratio of viscosity divided by surface tension. Now, and that leads ultimately to the drop snapping off. The question is, what happens when I have an indentation that's also flowing downstream? Well, it goes a certain length, a certain distance before it snaps off. But I know the velocity of the snap off, and if it's moving at this velocity, I can calculate the length that the jet has to exist before it breaks off. It's just this snap off velocity, that's the velocity in that direction, times the velocity here, the time it takes to snap off, times the velocity here. And if I calculate it, I get this ratio. It's the velocity downstream times this snap-off time. It's a very simple thing. Now I can very easily determine when do I go from forming a forming a drop right at the orifice to forming a drop downstream. Well, all I have to do is insist that the length of this jet is exactly equal to the size of the drop. That means that the drop grows exactly the right amount, so it snaps off right where it forms. And so all I do is I set this length equal to the radius done here, and that tells me that when this dimensionless number, the capillary number, the velocity downstream times the viscosity divided by the surface tension, when that's equal to 1, that's at that transition between dripping and jetting. There is another very interesting kind of instability. It's a similar instability. In this case, what I'm doing is I'm flowing the inner fluid very, very rapidly. If you look carefully, I hope you can see there's some small particles in it. You can see that they're flowing through the inner fluid very, very rapidly. And if you watch closely at the interface, you'll see all the hallmarks of an instability. If you measure, for example, the width of a neck, and you plot that as a function of time, you see there's an exponential growth. This depends on, because it depends on the velocity of the fluid, it depends on momentum. So this involves a different dimensionless number. It's the Weber number. It's not the balance between viscosity and surface tension, but instead it's the balance between inertia and surface tension. But we can plot everything together by just plotting and making a sort of a phase diagram, a state diagram of this transition between dripping and jetting. And it depends on the Weber number and the capillary number. High Weber numbers or high capillary numbers, you get jetting. In one case, you get these very small drops. In the other case, you get these very large drops. But still, you go between dripping and jetting. And when both numbers are small, you get the dripping regime. And in fact, you can perfectly describe the whole phenomenon by saying that there's a transition when the sum of these two dimensionless numbers is of order 1. Okay, so I've told you something about the physics, the simple physics. Again, it's a very simple description of the drop formation. But now let me show you really what's interesting about this, why it can be so useful. So we had another student, I called him Handy Andy Utada. He's very good with his hands. He really understood things. But he was a great student because, like all really good students, he made mistakes. And so he made something, we said, please all connect this together. And he made a mistake and he connected the wrong way, but he learned something really interesting and ultimately very important. So what did we do? Well, instead of connecting the two fluids like this, the way I showed you, he connected it the opposite way. He had the outer fluid flowing the same direction, but he connected the inner fluid to the other side. In this case, everything is collected through this small orifice rather than infused out through the orifice. It's infused in. This actually still works. It's nothing wrong with it. In this case, the outer fluid is coming around, but now it's hydrodynamically focusing. There's a viscous focusing of the inner fluid into a very, very small flow. This is actually even better because the diameter of the flow of the inner fluid is even smaller. It's not set by the orifice because you also have to have room to allow the outer fluid to go through as well. This works very nicely. Here you are in this dripping regime. In this case now, the inner fluid is coming from this direction. The outer fluid is looping around from way over here. It's looping around and it's forcing the inner fluid to be hydrodynamically focused. You can see now the diameter of the flow is much smaller than the diameter of the orifice of the collection capillary. You can do dripping, you can also do jetting. You can barely see the fact that there's a collection tube, there's a second orifice here, except because the index of refraction of the two fluids is exactly the same as the index of refraction of the glass, but you can see there's a little piece of dust, so you can see the fact that you have this jet form. It works exactly the same way. But what's interesting about this, this new kind of geometry, which we call, by the way, we call it flow focusing because this fluid focuses the anode. What's interesting is you have a lot of space here. So what you can do is just add a second tube. And now you have a third fluid. You have an inner fluid, you have a middle fluid, and you have outer fluid. And you're still doing this hydrodynamic focusing, but instead of focusing a single fluid, you're focusing a coaxial flow of two fluids, an outer fluid surrounding the inner fluid. This works quite well. It's easy to assemble. Again, it's easy to assemble because the square capillary allows you to align the two cylindrical capillaries very easily. So you thread one in, you thread the second in, and this is what it looks like under a microscope. And now you can see you have perfect alignment of these two very small capillaries. And people like Anderson, when they get good at it, could really precisely adjust, maybe have it insert a little, have very, very precise adjustment of the capillaries where they meet. And it's very easy, again, to do. And this is what you get. This is, again, these three fluids. And now if you look carefully, you get exactly what you thought you would get. That is, you get three fluids. So they're now not in the shape of a single emulsion, but rather a core shell shape, a drop inside of a larger drop, a core shell structure. Maybe it's easier to see it in this case where I'm changing the flow rate of the fluids. And here you can see I have a very small flow rate on the inside, so I have a very small drop in a much larger drop. Here they're more or less uniform. Here they look exactly the same. You might even say, are they really a double emulsion? But if you look carefully, you see there's one fluid, two fluids, and three fluids. And if you look really carefully, you can still see that you have this core shell shape. So you have one drop inside of a second drop, and we call that a double emulsion, a drop inside of a drop. Already, this is a very useful structure. It's very useful for many different reasons, but let me show you one of them. If I imagine, for example, that I have an outer fluid of water, an inner fluid of water, and I use a solvent for my middle fluid. But inside that solvent, I, for example, put phospholipids, I put lipid molecules, I put surfactant molecules, I put any kind of amphiphilic molecule. Because this is an oil and this is water, the molecules will coat the interface, and they'll coat both interfaces. But then if the solvent can be evaporated, if it's slightly volatile, I can evaporate the solvent, and then I'm left with a bilayer structure, but what's on the inside and what's on the outside is completely different. So this is a very precise way of making encapsulation. And in fact, let me take a little digression here. We did this many years ago, but right now, the reason that we have all these Zoom meetings, the reason that we don't have in-person lectures and things like that is because of this great pandemic. The thing that's or the remarkable thing that's happened with this great pandemic is the development of vaccines, many of which are these mRNA vaccines. The mRNA vaccines absolutely depend on the development of structures essentially that look like this. They're liposomes. They're made with they're actually made with microfluidics or with the microfluidic technology. They're made with a more sophisticated technology, but the basic physics is more or less the same as this. It's encapsulating mRNA on the inside of a structure that looks like this. It's called the liposome if it's made with phospholipids. It looks very much like this. They're more sophisticated, and I'll show you examples of how you can make them more sophisticated. But this is really a key to all the delivery of the mRNA, which is essential for all the vaccines that we all have to take, or we all should be taking. Here's an example where we made some. These are made with diblock copolymers, but the physics is exactly the same. You can see these are very, very uniform. We call them now, because they're made of polymer, we call them polymersomes. They're made very, very uniform. These are a few hundred microns in dimension. This scale bars is 100 microns. You can see there's a little extra bulb here. This is a little extra polymer. you want to coat the, completely coat the interfaces, so you add a little excess of the polymer. And in this case, we encapsulated something. We encapsulated a dye, a fluorescent dye, and we look at the same image under fluorescence, you can see that it looks green. And in fact, you can come back months later and the dye is still there. And you also can see that the encapsulation structures are perfectly uniform in size. It's actually interesting to watch what happens as you evaporate the solvent. Here's a series of images. You can see the solvent is evaporating. Let me show you this image here. Whoops, sorry. This image here. This is what it looks like. This is the bilayer structure, and this is a bulb of evaporating solvent. And this bulb eventually, this little solvent ball will eventually evaporate completely. But notice right here, you can see there's a surface tension. There's a contact angle. Sorry, there's a contact angle here. That means there must be surface tension. There must be, in fact, what it tells you is this structure here, there must be an adhesive energy between the two layers of this, the two layers that make up the bilayer structure here. It must be adhesive. Otherwise, you could not have a true contact angle like that. So in fact, how can I show you if they're adhesive? If these are slightly adhesive, what's the implication for the structure itself? And I can show you that by asking what happens when I have the structure, it's here, what happens if I try to collapse the structure? Well, how do I collapse the structure? Well, one way is to take a sledgehammer and bang on it, to put a big hammer on this. Of course, that doesn't work very well for these small structures in water. So instead, what I do is I put an osmotic pressure. I put a high pressure here that sucks the water out on the inside. And let me show you what happens. You see what happens is you develop these buckles. You buckle the structure, and a fluid-like structure would never exhibit this kind of buckling. That tells you immediately that you have to have a thin, elastic shell. These polymerisome structures have to be a thin but elastic structure. In fact, you can make measurements, and this was done by a student of mine, Sujit Datta, working with my colleague, David Nelson, and David helped us interpret the data. You buckle them, you just measure the number of shells that are buckled as a function of the pressure that you're applying, and from this you can calculate the elastic energy of the shell. And notice the pressures that you're using, they're pressures of atmosphere, and you're using about 10 atmospheres. And he said 10 atmospheres, that's like 30 feet or 10 meters of water. That's a lot of pressure, but these are not unusual pressures when you're talking about osmotic pressures. Atmospheric pressures are typical of what you get for osmotic pressures. And so this proves to you that you have these elastic shells that you're forming. Now, the fact that you can crush them is actually very interesting. So let me show you again some images as I crush them. You see it's buckling, but over time, and here's some images about 10 minutes later, over time, I can completely crush them. So what happens with whatever's on the inside? Whatever on the inside, unless it's a massive molecule that has to be relatively small in concentration, everything else is driven out from the inside. So what I've done is not only have I encapsulated something very precisely, very solidly, because I have a solid shell, but I've showed you how you can release it. You can release it by just crushing the structure with an osmotic pressure. I can do something else. I don't have to crush the structure. I can also blow it apart. And the way to blow it apart is not to put a high pressure on the outside, but to put a low pressure on the outside and use the fact that there's a pressure on the inside. So if I put a low pressure on the outside, I can do that just by putting pure water, it has no osmotic pressure, and then the inside will cause it to blow apart. These are these same polymerisomes with the fluorescent dye, where I put it under a very low pressure. You can see a small hole develop, and the dye diffuses out. If I come back about a half an hour later, there's no fluorescence left. And in fact, I couldn't show you an image with fluorescence. I can only show you this bright field image. It's interesting to watch this and what happens. Watch this particular polymerisome as I put very, very low pressure. You see, first notice it's getting bigger, water's rushing in, and then a hole breaks, it blows itself apart. and a hole breaks and it starts to shrink away. In this case, if you watch this one, you see that it gets larger, it always gets larger. Then you see snap, it gets smaller, and this one completely disappears. It just, everything on the inside diffuses out. So there are two ways of controlling the release. Not only can you encapsulate something very well, but you can control the release just by controlling the osmotic pressure. Now you can make a lot of other things. You're limited really in a sense by your imagination. You're just mixing fluids together. Here we've made solid shells. Instead of evaporating the solvent, we polymerize it. We use a UV curable resin as the shell. We polymerize it. We have solid shells. And now to break them open, you have to really crush them. We put them between two glass slides and you crush them. You get these Pac-Man-like structures here where you've built, made a hole. You can do a lot of other things. Here's something that's a very simple way. We made what we call a melt emulsification. We used a low temperature melting wax. This is a triglyceride. Its melting point is somewhere around 30 degrees, 35 degrees, something like that. So we just heat it up. It's above its melting point. We make the emulsions the way we normally do. Then when we cool it down, we get these solid wax-like shells that are very stable and very useful. And these then, to release them, you just have to heat them up. Here's an example. These are some colloidal particles. These are some dye. I'm going to show you images as I heat the shells up. I heat them up till they melt, and you see everything gets released when they melt, and the shells melt, they just break apart. This is actually something very useful. For example, if you want to do a drug delivery, we develop these things for many practical purposes. Probably the most important application is for drug delivery. We see that now in the current pandemic. This is one thing that we thought about, because we have a shell that's hydrophobic, a core that's hydrophilic. We can mix two different kinds of drugs. One hydrophilic drug that's denoted here by our prototypic model, which is just some red dye, and a hydrophobic drug in the shell, which is like an oil where we put green dye. And very often you want to deliver drugs, you want to deliver more than one drug because they work synergistically. Two drugs work better than each of the individual drugs. However, if that's the case, you want to deliver to the same location exactly the right ratio, as well as the right concentration of the drugs. The ratio has to be right. Because you're delivering locally the same relative amount of each of the drugs, this is a very efficient way of doing it. And this is just an example. Here we have our prototypic, our model drugs, the two dyes. We heat them up. And when you get above the melting temperature, you see both dyes are released exactly the same point, exactly the same way. And we did some simple testing. We used some synergistic drugs. We put them, we used some cells that were some cancer cells. We used some anti-cancer drugs. And we showed that they work much more efficiently when we had the combination of the drugs than when we had single drugs. And we could deliver them very effectively to exactly the same point. Well, okay, Liang Yin Chu, one of our early visitors from China, came and he said, look, Andy already made some devices. What could he do differently? He made a slightly different geometry. Instead of having spontaneous emulsification to make double emulsions, he broke it apart. He took a collection tube, so he took a second capillary tube. He used the first capillary to make drops, one type of drops, and he used the second capillary to collect those drops into a second droplet. So here you watch, here you see one drop come into the second drop. I've watched this movie many times, so I know it'll take a little bit of time, and eventually we'll see, here comes a second drop, and now I don't have to wait so long, so I'll wait. A third drop will come, and then it'll snap off, and I'll have 3 drops inside of one. So this is now an opportunity to make what I call designer emulsions. These are emulsions where not only do you make a core shell emulsion, but you can make different numbers of drops on the inside. So you say you want a core shell emulsion. I say, well, how many drops you want? Oh, you want 5. Okay, well, here's 5. How big do you want the drops? You can control very precisely, just by controlling the flow rate, how many drops and how big you make the drops. But Lang Yin showed that to me. I said, well, look, we already make double emulsions. What else can we do? So he said, look, I have an emulsion generator here, an emulsion generator here. What about this? So he just added another emulsion generator. Now you're making drops that go into drops that go into drops, and you make what I call the Russian dolls of drops. You make drops inside of drops inside of drops, again, with this very exquisite, very precise control. Of course, if you do this, you might say, well, this is very sort of academic exercise, making drops inside of drops inside of drops with this really precise control. What kind of useful thing can you make? So here, Lang Yin tried to do something to show that it's actually useful. What he did was he took a water drop in oil, so it's a water and oil emulsion. Inside this water drop, he has some very, very delicate peptides, and they're in this oil continuous emulsion. And because it's an emulsion, there's always a lot of surfactant around. The trouble is that the surfactant will denature the peptides. So you want to protect the peptides. So what he did was he used the ability to make these multiple emulsions to put a shell that's a hydrogel. This absolutely protects what's on the inside. But this particular hydrogel is thermosensitive. That is, as you heat it up, it shrinks. So let me show you what happens. I'll show you some images as you heat them up. It starts to shrink. It tries to compress the inside. It's incompressible. So instead, it just tears itself apart and it releases the drops. So what have I showed you? I've showed you a way that you can make a very robust encapsulation structure, but a precisely controlled release structure. And if you think about encapsulation and release of materials, it's very difficult to do something that both encapsulates robustly and releases controllably. It's easy to make something that's very robust encapsulation, but it's very difficult to cause it to release. Similarly, if you have something that releases very easily, it's very difficult to encapsulate it robustly. But here, because you have this tremendous precise control over the fluids, your imagination can go wild. You can make all these different kinds of structures where you can do both. And Lang Yin went back to his home university, Sichuan University in Chengdu in China, and he developed even more sophisticated structures that allowed him to make these Russian doll kinds of emulsions where you don't have just single types of drops on the inside, but you have multiple drops on the inside. Again, just following on with this concept of just mixing the drops together using these capillary devices to control the fluid. So I hope I've showed you that these are very interesting structures you can make, but I also challenge you to think and to ask, can you make enough of these things? You're making them one by one. They're very highly reproducible, but can you make enough? And so the question is the process scalable? And I asked Anderson, I told you he makes his device in 30 minutes. So I said, okay, Anderson, we really need to test how good the devices are. Please make 2 devices and let's compare them. So he went and an hour later, he came back, 2 devices, and we tested them. They were perfect. I said, oh, that's good. Now make 10 devices. So he looked at me, a little funny look on his face. He went away, and about a day later, 10 devices, half hour each, that's 5 hours, got to have something to eat, get some sleep, come back the next day. He had 10 devices, and again, we tested them, and you could not distinguish one from the other. All the drops were exactly the same. So then I said, okay, Anderson, I want to make some rheological measurements. I need a lot of material. Can you make me 10,000 devices? So he looked at me, he smiled, and he walked out of my office, and he graduated. Who wants to make 10,000 devices? You have to do something else. You have to somehow parallelize them to make many, many devices. So Xinyun came along, and he made multiple devices like this, so he could make them a little faster, and that worked. But really, the way to ultimately do it is to go to the other kind of technology, move back away from our capillary devices, go to the more traditional devices. So if you're at Harvard, you go across the street where George Weitz is, and you tell him, George, please show us how to make things with soft lithography. And I won't go through the details of soft lithography, but what it is, but this is a more traditional way that people make microfluidic devices. And the important thing is that to define the shape, you use a mask, and that makes a pattern in a photoresist. So now if you want to have one device, you very carefully design this one device. If you want to have two devices, you just copy paste. You take the mask and you make a copy of it. 4 devices is copy paste, copy paste. 8 devices, copy paste, copy paste, copy. You see the scaling is completely different. So these kind of devices, you really can scale in a different way. Here's an example. This is one of the device that makes double emulsions. This is just showing each movie as a different flow rate, just to show that you can do what Liang Yin did. You can make these designer emulsions, 1, 2, whatever number of drops you want. You can stack them together. So these are now triple emulsions. If you look carefully at the flow here, you see there's a drop inside of a drop inside of a drop, always a perfect emulsion. Adam Abate, the person who did this, said, well, Yang Yin already made triple emulsions. I want to just show how easy it is. So he made a quintuple emulsion, a drop inside of a drop, inside of a drop, inside of a drop, inside of a drop. It's really easy. You just stack them all together. Turns out there's some interesting physics. You always just get a single drop in each of these, in each case. That has to do with the crowding effects, but it's not so important. It just shows that you can do it very easily. Of course, that's not what you want to do. You don't want to make multiple emulsions, multiple emulsions. You want to make many of them. So what you want to do is parallelize and stack them and ultimately go into 3 dimensions. And so Mark Romanowski, another graduate student, he came to me once. He said, look, I've been working for 4 years. I don't have any results. Do you have something that I could do that would give me some results very easily? And so I found out what he did, and he said he worked in the clean room. I said, well, look, I'll give you a challenge. I'll give you a liter of material. So I'll give you a device that's 10 cubic centimeters, 10 centimeters by 10 centimeters, 10 centimeters. That's a liter. I'll give you a device that's large. I'll give you 2 pumps, and I want you to make a liter of material of internal phase per hour. So I said, if you can do this, you can write a paper and people will really be interested in the paper. He wrote a paper about his results, and it was the most downloaded paper of the month when he published it. I said, the other thing is you can graduate. And a year later, he graduated. So this is his device. He made 25 emulsion generators. These are double emulsion generators now, so it's much more sophisticated. He made 25 of them in a single device. And let's see, let me just play this again. If you watch carefully here, as I watch the movie, and right here, you see eventually you start collecting a lot of material. Esther Armstead came along and she said, well, I want to do much better than what Mark did. So she made what we call the millipede device. It looks like this, and there's 500 emulsion generators on each side. You can see each of these has a channel with 500 channels on this side and 500 channels on this side. This is what it looks like in operation. And you can see now you're making large number of drops. A lot of interesting physics here. I won't go through the physics. You can change the size just by changing the dimension of this. You make very, very uniform drops. But now what's interesting is the numbers. You go through the numbers. The highest flow rate that Esther achieved before she left, she's done much higher flow rates since then, but before she left, in one device, 1 millipede device, she could make 25 milliliters per hour. The size of the device was 4 by 1 by 1/2 centimeters. I'm not going to do any fancy organization. I'm just going to say, well, here's my challenge. I'm giving you 10 by 10 by 10 centimeters. In that, I would be able to fit 400 devices. So with 400 devices at 25 milliliters per hour per device, you're making 10 liters per hour in a liter of material. So you have a whole device as a liter, you're making 10 liters an hour. Is that useful? The answer is actually it is. So to show that, we started a small company to use microfluidics to make materials. Of course, you can make lots of materials, but you wanted to make something that's very high value added. You could make drugs. This was long before the pandemic. We weren't thinking about that. We wanted to get something that we could get to market, not in an emergency case, but an actual case very quickly. Drugs take a long time, but cosmetics is a high value added product that doesn't take so long to get to market. And so in fact, we use this device to create some structures that were sold. This is sold by Amori Pacific, a company in Pacific, in Korea, sorry. They sold the device. Here's an example of a commercial device that they're using to make it. This is what it made. It's sold by La Prairie, a very high-end cosmetics company. They made something they call skin caviar liquid lift. It's basically structures that look like this. This was on the cover of CNE News. And I told you that it's very high value added. If you look at this website here and ask, what does the next page of the website say? You can see that it's 500 USD for a small container of 50 milliliters. People will pay a lot for high-end cosmetics. In fact, they made a different structure. This is another version of a double emulsion generator that was a product that was introduced about 5 years ago. This is the product, Hydra Beauty Micro Serum. It was introduced by Chanel. And in the first 9 months, Chanel produced 1 million units. Well, Capsum produced for Chanel 1 million units in 9 months. They weren't operating 24/7. They just had one machine size of a desktop. So this shows quite positively that you can produce enough material not only to be useful, but actually be commercially valuable. Okay, let me spend just a couple of minutes and show you the other general use of microfluidics. The more common use is what I call lab-on-a-chip applications. That is, you miniaturize a whole laboratory onto the scale of a chip. And I'm going to show you that you can do really interesting work with that, again, using drops. And the difference here, the reason that we use drops is the same reason that we use the drops for materials, that is, you can encapsulate something. But in this case, what we do is we encapsulate an experiment. So if I want to do lots of experiments, now I can use a robot that uses many pipettes and works with what's called a microtiter plate. This is a 384 well plate. There's 384 wells in this plate. There's maybe 10 to 100 microliters in each well, and each well is a separate experiment. What if I want to do many experiments? Well, I can replace each of these wells by a small drop. This is the same thing that I showed you before. These drops are now very uniform, but the size is very different. The size, instead of being 10 microliters, the size of these is about a picoliter. That's 8 orders of magnitude difference. So each of these drops I'm using now not to encapsulate something, but I'm using as a reactor. I'm using it like a test tube. But the size of the test tube is much, much smaller. Why is that important? Well, so let me just remind you that the size that I'm going to talk about a 10 micron drop, that has about a picoliter, and I'm using the drops at 1,000 per second, I can do many, many experiments. So here's a simple kind of argument why that might be important. Using robots is something that's become very popular for doing screening. For example, drug companies, they may have, when they make a drug, they store every drug they make, and they build up these library of drugs. And then if they find some new biomarker, the first thing they do to look for a drug is not try and develop a new drug, but ask, do any of the drugs work? So they might do a screen against all the drugs. So this I did about 10 years ago. And then a typical library of drugs was about 100,000 compounds. Now most drug companies have more like 2 million compounds or more. And if you want to measure a binding constant, you might want to measure 10 concentrations to measure a true binding constant. So that would mean you have to do 1 million experiments. You can do that with the robots, but let's say you use a small amount. This is just using 1 drop at a time. You use 10 microliters per experiment, but you do 1 million experiments. It's still 10 liters of reagent. That's very, very expensive. Ultimately, that's what limits how many experiments you do. And I once calculated, if I do a few pipettes, I do say 8 at a time, I do a few pipettes for each reaction, it would take me 4 months running 24/7 with a typical low-end robot, which is what I might have in my lab. But if I do it with microfluidics, where I use a picoliter per test, 1,000 tests per second, I can do the whole measurement with just 1 microliter of reagent. And I can do it in about 20 minutes. You can go have a cup of tea, come back, and your experiment is done. In order to do this, I have to be able to control the drops very precisely. So we built chips that control them. Here we're making drops. We can split them. Here we're splitting them into 2 equal sized drops, 2 unequal sized drops. We can split them many times. Here we're splitting them 1, 2, 3. We're taking large drops, making small drops. We can take a little bit off, so we can do assays. We can just take a little test. Here we're taking about 1% of each drop and we're analyzing it downstream. We can mix drops. Here we're taking pairs of drops, so you have reactant A, reactant B, and 2 different drops. We bring them close to these electrodes. We put an electric field on, and it merges the drops. This has led to a more recent development, which we call the pico injector. Here we're injecting some dyed black fluid into each drop as it goes by. We're literally putting a picoliter of fluid into each picoliter drop as it goes by. This is controlled by the electric field, so we can have multiple pico injectors. You can see only this one is injecting, the other 2 aren't, but we just control the field so we can mix and match, have different injectors. We can detect things. We tend to use fluorescence, but you can use anything that you can see. Fluorescence is very convenient. And we can sort things. If you really want to do an experiment, you want to select one from many. So here we're taking 2 different colored drops, we're spacing them out, and you can see we're using, again, some electric field. In this case, we use a gradient of the field to put a force on the drop, and every undyed drop is pulled to the bottom. All the others go to the top, the lower resistance, so the weights. So here's just a very simple example. I just want to show you that you can do many, many things. Here's one example that takes advantage of being able to do large number of experiments with very low amount of material. Imagine you have an enzyme. Here we have a very common marker enzyme, horseradish peroxidase, HRP, very widely used, very efficient. Nature has made it very well. Why it's so efficient is that it takes its substrate, amplex rad, in the presence of peroxidase, it turns it into a fluorescent product, a resirufrin, which you can see. And so we want to know, can we improve on that? But look, an enzyme is very complicated and nature has made it really very good. How can you improve on it? Well, rather than trying to do the chemistry, you can do what nature does. You can do evolution. And so what we do is called directed evolution. Jeremy Agresti did it. The concept is very simple. You start with the parent gene, the gene that codes for the enzyme, and you make lots of mutations. Yeah, you have the delta mutation, all the mutations of these RNA viruses. Same thing here. You take the DNA, you make mutations, and you make a library of mutated genes. Then you make enzymes of it. We use a technique called a yeast surface display. We work with Dane Wittrup at MIT. Basically, you take a plasmid, you take the mutated enzyme, you put it into a yeast cell, and it makes many copies of the enzyme on the surface. Here's just showing what happens. These are the drops that you put the yeast cell in the drops. You can see that the drops that have the yeast cells turn fluorescent because we've added the substrate. It becomes fluorescent. So now we do this simple experiment, taking advantage of all these tools that we've built. You make a library of mutated genes. You put 1 gene per yeast cell, 1 yeast cell per drop. You add the substrate and you ask, does it fluoresce? Most of them won't because you've mutated the gene. You've made the enzyme not work. Only a few of them will actually fluoresce. And you take those out, you grow them up, and you can do multiple rounds of selection because the yeast that are producing it are still alive. And when you do that, and you just measure the average fluorescence, you see that when you do 1 round of mutations, after a few rounds of selection, you see that you begin to get higher level of fluorescence. This is the average fluorescence of the parent type. You begin to get a few higher level. You go and you look and you see that you've developed a few enzymes that have mutations that have slightly higher fluorescence. They're not much higher, they're just a little higher. And roughly speaking, we found 100 unique enzymes, but we started with 10 to the 7. So we found 1 and 10 to the 5 work roughly as well as the parent type. But then what we did was we took those ones, we took the top 20 of them, and we mutated them again. And then we immediately found things that worked really well. And how can I understand that? In a very simple way, this is a fitness diagram. It's like an energy diagram, but upside down. So I'm already on a peak that works really well, and I want to go to another peak. nature's put me on this peak. I want to go here. And this is fitness space. So this is mutation space. So problem is, if I mutate things, usually I fall into this deep valley. If I want to go here, I have to, instead of jumping from one to the other, because if I take a jump, it's very unlikely that I'll land here. Instead, we did what was called a round of neutral selection. We found something that worked almost as well. And then we did a second round of selection. And this is actually something that you can't normally try because you're only getting 1 in 10 to the 5. But we can do this very easily. In fact, just to show you how well this works, I compare it to trying to do it with a robot. If I did it with a robot, I need something like, we did something like 10 to the 8 reactions. If we were using the normal amount of reagents, it would have been 5,000 liters of reagent. For us, we used 150 microliters. It would have taken about 2 years to do it with a robot. We took about 7 hours just for the screening. Adding everything out with the robot would have cost about $16 million. This was calculated about 10 years ago. There's been inflation, so it's quite a bit more. We estimated that we spent about $2.50. So this is a very, very efficient way of doing this. In fact, I showed this to one of my colleagues, David Nelson. He has a good sense of humor. He said, oh, the remarkable thing is you don't pay your students very much because they work for 7 hours and only cost $2.50. So I assured him that didn't include the student labor and that we paid our students much better than that. Okay, I think I've talked long enough. I apologize for going a little over. I hope I've shown you some of the beauty and the magic of microfluidics, both its utility for making new materials, the fact that you can scale up the production so you shouldn't be afraid of it, and the fact that you have all these different kinds of uses. And I didn't really hint about all the different types of uses that you have in lab-on-a-chip applications. And with that, let me thank you for your attention, and I'm happy to stay on to ask any questions. I apologize if I've gone a little over.
Thank you, Professor Weitz, for a highly motivating talk. I'm sure the audience will greatly benefit. I see there are many interesting questions already in the chat box. So I would just maybe start with, so there's a question of stability of drops inside compound emulsions. So the question is, do you see the drops moving and coming out of the outer drop? And what control that?
Yeah, you don't necessarily see them come out, but they may merge and they may coalesce. And that's a very good question. It's a very, very important question. And the way you control that is by proper use of surfactants and stabilization. That's a non-trivial thing. It's a very difficult, a very challenging thing to do, but it's something that people who've worked with emulsions have done for many years. And you use all the tricks that you can use with traditionally made emulsions. You use the same tricks here, the proper choice of surfactant, the proper choice of gelation. There are many different ways of doing it, but you have to control the stability of the interfaces.
Yeah, the other question is about drop coalescence when these drops are coming and they're merging when they meet. So the question is, what controls the coalescence? Is there any surfactant involved?
Yeah, again, that's an excellent question. I didn't discuss the surfactants. You absolutely need to use some kind of surfactant. There are many types of surfactants that you can use, but the whole role of a surfactant in the case of an emulsion is to ensure stability. That's a challenge, but it's something that has been solved in many cases for emulsions. Emulsions are very stable, or you can make very stable emulsions. You have to do the same thing here to create the to create stable emulsions.
There is a question regarding the oscillation of these the expanding structure in the jet when you talked about absolute instability.
Yes.
What causes these oscillations, and
So that's a very good question, and that instability is its own instability. It's called a stationary instability. That occurs because you're flowing things so fast that you're blowing up the drop while the actual instability is remaining more or less stationary in space. But once the instability goes, it's truly an instability. But a normal instability, if it's highly unstable, will immediately lead to snap up. It'll just grow, or in this case, shrink to zero right away. In the case of the oscillatory instability, you're driving something, you're making it unstable, and as with all instabilities, is something that's slightly unstable. So there's a restoring force, the driving force. The driving force is a little over the restoring force, and you get an oscillation that exponentially grows. And that's what you're seeing with that instability.
Another question is, in case of non-Newtonian fluid, if you want to generate a drop of non-Newtonian fluid inside a Newtonian fluid, or vice versa, whether the regime diagram would change?
Yeah, I mean, the state diagram may change a bit, but not by that much. It turns out we've done a lot of work with non-Newtonian fluids, and there's maybe some elongational forces, some additional forces, but if you are in the limit where these hydrodynamic instabilities are controlling it, it's controlled by surface tension, and the surface tension forces have to be large enough compared to all the other forces that you form a drop. So typically, the non-Newtonian nature of the fluid does not lead to something that overcomes surface tension. In fact, what often is a problem, if you make the fluids too viscous, it just takes so long that it's very difficult to actually generate the drop. And so the dynamics is what creates more problems than the non-Newtonian aspects.
Yeah, another question on this drop within drop within drop. We need to release the inner drop. While we need to break the outer cell while keeping the inner drop intact. So, has any application identified of this?
Well, we showed that prototypic example where you might have an emulsion that's a very delicate emulsion, can't withstand the vagaries of the environment for very long. But if you could release it and use it right away, that would be something of great value. And it turns out, I hate to say it, but I've learned a lot more about cosmetics than I ever knew before. And the key about cosmetics, one of the important things that the consumer wants is how it feels on the skin. And for that, you want, for example, it feels very good if you make something that remains as an emulsion until, boom, it just breaks and you get a completely different feel to the skin. The serum that Campson made for Chanel is exactly like that. And that had a very different skin feel. And it also was made, it was an emulsion made without using surfactant, which has a very good, many advantages for cosmetics. Surfactant tends to make your skin itchy and dry, whereas you want to moisturize it, you'd really rather not have surfactant. By using these microfluidic technologies, you could make these drops that had different feel, different skin feel, and different, and could be stabilized without using surfactant. You just can't do it in other ways, and that's just a practical use of this technology.
There's another question about this jet breakup, as the location of the breakup was changing with time. The question is, can it be predicted theoretically, or can it be?
The answer is yes and no. It's a stochastic effect. It's basically due to the fact that the instability that's causing the breakup is the growth rate as a function of wave vector is fairly flat. So it doesn't grow at a very specified wavelength. Almost any wavelength will become unstable. And so it's just random fluctuations that lead to the particular wavelength that lead to the particular size. If you know what the size of that fluctuation, you can calculate the size of the drop, but that's a stochastic event. So you can do it on a probabilistic calculation. You can't do a deterministic calculation.
Okay, maybe a few more questions, couple of more questions. So the next question is about in this drop inside drop, how do we perceive the role of RP instability in terms of either dripping or jetting? Is it going to be the same mode of drop formation when we talk about?
Good question. And the answer is that it depends. It really depends on exactly the geometry and the flow rate. Sometimes you'll have dripping in one case, jetting in the other case, vice versa, both dripping, both jetting. So you can control both of them. And it's the fact that, in fact, that you don't have the same precise control that means that it's actually difficult to use that. That's the reason for going to the other geometry, where you had more precise control of each of the flow rates, in which case you went much more to a dripping in each case. But more normally in the spontaneous emulsification of multiple drops, you can get both formations.
Right. So the other question is about satellite drops. Do you get satellite drops, and how do they affect your yield?
Well, you do get satellite drops very often. That's a good question. You often get satellite drops. I don't know if I showed any in these images, but you often see them. And typically what we do is we just don't worry about them. They're tiny compared to the size of the drops. There's actually, if you think about it, the volume of a satellite drop, because its radius is so much smaller, everything scales the cube of the radius. The volume is very, very low. So there's not very much material in it. So we typically just don't worry about it. But there's no question that you get them. Ideally, you'd like not to.
Maybe only two more questions. So this is about when you are pulling this glass capillary, what kind of temperature would be required? Maybe you're worried about cracking and so on.
Yeah, basically, you want to work close to but below the glass transition. So the the glass starts to flow a bit, but not too much. So I don't know, several, 1500 C, something of that order, several hundred C to somewhat below 2000 C.
Yeah.
This is all done by machines now. You can go to your friends in biology, people who do patch clamping. They for years have used these kind of capillaries. They pull them, so you can just buy a machine that pulls them, and it does in a very reproducible way, a very controllable way, and you just have to dial in what you want more than anything else.
Probably the last question. So this is about, I think you showed two different configurations for drop generation initially, one flow focusing, and initially, it was the first version, and then the second version, which your student did it by mistake in the opposite direction. So how do they compare in terms of drop generation characteristic?
Well, they're more or less similar. I guess the advantage of the flow focusing is that you get smaller drops for a given orifice size because you have to force both fluids through it. It's maybe a little bit harder to adjust. You have to get the flow right, but it's not so difficult. And so it's maybe a little preferable to use the flow focusing.
Okay. Or so we can go offline now.
Okay. Well, thank you very much.
Thank you. Thank you very much,
Very exciting.
for doing this for us.